Okay, hello everybody. Okay, so before we head on to look at the content and the questions, right? So just a quick introduction. I do not want us to be here just listening to a voice speaking, just hearing a person speaking out here, right? We are here to learn together here. So I would love, absolutely love to hear your voices, asking me questions and answering my questions as well. Right, we are not here to just listen to a video recording of our friends at home, not here to just watch a movie of a person speaking. So please learn with me, right? Your nods, your shakes here for yes and no's, much appreciated. Can? Okay, so first and foremost, a little bit about myself. Right, you can call me Mr. Jason, Mr. Pang, teacher, whichever you're more comfortable with. I've been teaching for about 10 years or so. I hope I do not look like it. Okay, right. But I studied life sciences in NUS, which was just the university's way, fancy way of naming biology. All right, B for biology, which is the best science, as you will come to see later on. Okay, right. So I dabbled a little bit in psychology, which was just more about learning about the human mind. Because the brain is an amazing organ, and by the very fact that I'm saying this, the brain is complementing itself. All right. Okay. So just gotta think about how bio can actually further let us understand and appreciate our own bodies and the world we live in. Right. So this is what I had the opportunity to learn together here. And outside the classroom, I'm not just a boring person speaking about bio over here. I love being outdoors. All right. So this picture was taken when I was hiking in Australia, the Blue Mountains. Absolutely wonderful place to hike at. And I also do a little bit of climbing, right? As you can see on the right hand side over there. And I have the wonderful honor of combining my passions for climbing and teaching. So I'm also a climbing instructor, as you can see in some of the pictures over there, right? And right smack in the middle, other than being out in the mountains or on the wall, I like being in the hall as well because I make music. So I play in a band as well. In fact, we just had a concert on Sunday. Yeah. Okay. So if I do see any of you in the hall making music or on the wall climbing or in the classroom learning with you more in future, do say hi. But this is just a little bit about me over here. So it is not just a person speaking. I'm not old. I'm not boring old man. Right. So please learn along with me here. Ask me questions. Answer my questions. Nods and shakes greatly appreciated. Okay. So we are over here. Today is the brand spanking new center. Today is literally the second day we are using this classroom. So wonderful place to learn together with you. Very, very near to the Tampines Malls over there. And yeah, just a quick introduction to the center only. Okay, we'll talk more about this a little bit later on. Let's get into it. Okay. But before I begin, can you help me to close the door, please? Thank you. Okay, so just making sure that our friends at home are also looking at the correct handouts. Okay. All right, so first things first. How many have show of hands? How many have done the mock paper aside from like now? Okay, thank you for your efforts over there. Okay, so we're gonna be working on it a little bit later on, but allow me to walk you through some of the topics that I have selected over here. So in the physical copies that you have collected, right, you have all the topics for Sec 3, right? We do have a couple of Sec 4s here with me over here. Not to worry, we're gonna be looking at some of the topics here as well. Okay, so let's dive into it. The first topic that I want to look at together with you here will be for biological molecules. So please flip to biological molecules here with me. Okay, we are not going to be looking at every single topic. I definitely don't have time for that. Okay. Okay. Biological molecules is split into two components, the nutrients and the enzymes portion, right? Notably so, some of your schools may even teach it as you call it different topics. But typically in the syllabus document that the ministry set out for us, they're under the same topic. So with a slight overlap. Let's look at nutrients first. The first one being carbohydrates. Can y'all tell me if y'all use these three terms in school? Monosaccharides, disaccharides, and polysaccharides? Yes, yes, no? Some do, some don't, right? Okay, so let me share with you why I very much encourage us to be familiar with these terms. Mono, di, poly. I think everybody knows this. Correct or not? Mono, di, poly. One, two, many. Okay, but what is a saccharide? Now, saccharide is loosely translated as little sugar. So one little sugar, two little sugars, many little sugars. What is the little sugar that we are talking about? Continue to space. Then they don't have to squeeze so much here. Then you don't need to squeeze. Okay. Right. Okay. So first and foremost, the little sugars here, the simplest one being the single unit. Right. Quite obviously, two of them die, then many of them polys. I would actually like to start your memory by looking at the polysaccharides because you actually encountered starch already. Since primary school, you already know about starch already, isn't it? Okay. So over here, if you look at this entire frame here, wow, 9 different things. Do we have to remember all 9 of them? The answer is a resounding yes. Okay, but when we start over here, I really want to help us. Okay, I'm seeing some responses. Oh, so many things to remember. I really want us to try and move away from the idea that bio, just memorize can already, just read can already. Whenever I hear someone say that, my heart dies a little bit inside here. Bio is an amazing science to learn together. Why? Like I said again, it's about learning about yourself. Right? Learning about yourself, learning about how your body interacts with the world around you. Okay, but is it without memorization? That is rubbish. Nothing, you cannot learn anything without remembering anything. When you first learn how to walk, you must remember how to walk, right? Now need to think about it. Don't think what? And that's what we are going to try to get to. Okay, that's what we're going to try to get to. The more you understand how bio works, the more you appreciate how all these little things link together. You learn more by remembering less. How do we do that? For the polysaccharides, instead of remembering 3 different things, let's try and group them together. If I want to group starch and cellulose together, can someone tell me, how are starch and cellulose related and different from glycogen? How are these 2 things similar but different from glycogen? Absolutely, thank you. And your name is? Hillary, thank you, Hillary. So starch and cellulose are found in plants, whereas glycogen are found in animal cells. Okay, so the first comparison we can use is location, plant or animal cell. Okay, thank you, Hillary. Right, moving on to our next comparison. Now, now I want to put starch and glycogen together, which makes them different from cellulose. The word is storage. They are not stored, they are the storage forms. You see the slight difference there? Thank you. Your name is? Brandon. Sorry, starch and glycogen are storage carbohydrates, right? One for plants, one for animals. Cellulose leh. What is it for? Cell wall. Thank you. Provide structural support. Okay, provide structural support. So immediately we have 3 things here. We can separate them according to location and function. Plant versus animal, support versus store. Okay? Okay, so moving on. We're going to group our monos and disaccharides together. It is at this opportunity that I want to ostracize our dear sucrose. Why is sucrose being ostracized over here? Sucrose, of the 6 things over here, is the only what? Non-reducing sugar. Thank you. Non-reducing sugar. Sucrose over here is a non non-reducing sugar. Thank you for your answers over there. Right? Now, very fortunately for our syllabus, we do not need to explain what it means to be a reducing or non-reducing sugar. That is a story for next time, okay? For our syllabus, we just need to be able to identify you reducing, you non-reducing sugar. Right? This brings us to our first test in this syllabus, isn't it? It's a what test for reducing sugar? Benedict's test. Thank you. Benedict's test for reducing sugar. So, this means that the 5 circled in this green thing are the reducing sugars. They are the reducing sugars. Okay? Monosaccharides and disaccharides are the sugars. Then with that in mind, what will the observation be for the polysaccharides over here when I conduct the Benedict's test for reducing sugars? What colors will I see? Blue, because the polysaccharides, they are not sugars at all. Think about the last time you had a mouthful of white rice, plain white rice. No soy sauce, no sweet sauce, nothing. The white rice sweet or no? No ma. Okay, so starch, not sweet. Use that to help you to remember the polysaccharides are not considered sugars and hence blue color with Benedict solution. Okay? Okay? Benedict test for reducing sugars. So that's emphasis here. Must be a sugar. Sucrose is a sugar. In fact, sucrose is the most common sugar. The sugar that you add into your cakes, the sugar that you eat with your roti prata is sucrose. Okay, so sucrose is non-reducing sugar. The rest are all reducing sugars. So how do we link more things together such that we remember less of them? Now, monosaccharides, disaccharides, one little sugar, two little sugars. Wow, how to remember? How are they going to come together to form our three different disaccharides? Okay, we're going to list down our disaccharides first. We're going to list them down in this order. We have maltose, we have lactose, we have sucrose. Okay, so they are all disaccharides, meaning they're going to be made up of two monosaccharides. Instead of having you remember 6 blanks, do you see? Instead of remembering 6 blanks, it will immediately cut down your job by half. Why? All your disaccharides have at least one glucose. That's it. Immediately, you remember half the things that you need to. Can you see that coming together? Okay, disaccharides, or at least one glucose. Okay, next step. Maltose, just one thing only. Simplest disaccharide. Maltose, the simplest disaccharide. What can be simpler than having another glucose? Okay, what can be simpler than just having another glucose? Huh? Now, the next one, I don't know whether to say it's even simpler or not because it is in the name. Lactose is when glucose plus galactose. And awesome. Now, the last one. A couple of years back when I was teaching this, suddenly my student from the back shouted at me, Mr. Bang, sweet girlfriend. Why suddenly sweet girlfriend? SGF. Okay, so sucrose is the thing that makes it sweet, right? So we're looking at sucrose, glucose, and fructose. That's it. Okay, right? So right from the top again, we have the simplest disaccharide. We have lactose from galactose. And then what's left? Only got fructose left. Can? Okay, so as you can see, we have glucose definitely present there first. And then we list them down like that. Can? All right. So why is this important? Because for pure bio, you need to know that all my monosaccharides, right? All my monosaccharides have the exact same chemical formula. All my monosaccharides have the exact same chemical formula. Hey, how can they have the exact same things, but they are different things? Someone help me make sense of this, please. Arrangement, thank you. Right, so the 6 carbons, 12 hydrogens, and 6 oxygens are all arranged differently. Just like if I give all of you the exact same Lego box, you all can build different things, huh? Fair? Can? Okay, so that's the arrangement there. So likewise, for disaccharides, they also have the same chemical formula. Yes, Elizabeth, I'll get to that. So disaccharides, want to make a guess? Formula of disaccharides. Mono is 1, then di is 2, right? So what will you do to the chemical formula? You just have to times 2, but you must remove something. Let me write this down. You tell me what I remove. This is the formula of all the disaccharides. What did I remove? H2O. Thank you. Your name is? VJ, right? Remove the H2O. This is actually a byproduct of the reaction. When two monosaccharides come together, a water molecule must be removed for the bond to be formed. Can? We do not need to know the name of the bond. I need you to recognize this one only. Can? Okay? Even if you, very simple, even if I never teach you, your school never teach you, are you able to come up with this? You just times 2 minus water. That's it. Okay? However, polysaccharides, no chemical formula. Because why? Poly just means many. How many? I don't know. Okay? However, if I were to share this with you, What can we derive from this? What about polysaccharides? There's something very unique about them. All the polysaccharides are made up of the same thing. They are all made up of the same thing. All the polysaccharides, no matter starch, cellulose, or glycogen, they are all made up of glucose. So the same question again. Hey, if they are all made up of glucose, why are they different things? It's the same answer. The glucose molecules are arranged differently. Okay, they're all arranged differently. Okay, so have that image in your head. I'll talk more about that later on. Okay, so over here, elements present, carbon, hydrogen, oxygen. Now, the ratio, very important. In identifying your carbohydrate, H and O are always present in a 2:1 ratio. How does this make sense? I would actually use the name. Look at the name itself, literally the name itself. Carbo tells me carbon is present. What does hydrate mean? To hydrate is to drink water. Water is present. Literally in the name itself can help you to remember what elements are present in carbohydrates. C, H, O. That's it. Is there something special about the H and O? Absolutely. The H and O's will always be present in a ratio of 2:1, in the ratio of water. In the ratio of water. And that's why they are called carbohydrates. All right. If you look at the monosaccharide, did I follow H2O? Is the ratio H2O? Yeah. Is the ratio H2O over here? Yes. And quite obviously, this one will also be kept. Okay? So first thing, only have CH and O. Next thing, H and O 2:1. Okay, next thing, H and O 2:1. So therefore, this is what we need to remember to understand whether a molecule is carbohydrate or not. So how will this concept come into play? Question give you some random C something, H something, O something. You count lah. If the H and O are in the ratio of water, carbohydrate. Okay? Okay? Awesome. Right, so function, what do we use carbohydrates for? No matter what kind of carbohydrate we take upon the mental of a humans, what do humans do with carbohydrates? No matter carbohydrate, what form you consume, all of them, as it goes through the digestive system that we'll look at later on, it will ultimately be broken down into its simplest one, glucose. All right, the two main functions, uses of glucose will be for cellular respiration to release energy, and also to build up some other stuff that the body does not have enough of, like your lipids or your amino acids. Fairly straightforward. Okay? Moving on to the bottom, we have the Benedict's test, reducing sugars. Please remember your colors over there, very important. Why is there a need for the hot water bath? Two questions here. Why must hot water bath? Why cannot Bunsen burner? Remove impurities. Not quite. Good try. Thank you. Not quite. What is the reason for heating up the reaction? Why do I need to heat up the reaction? Absolutely. That's it. Okay. I heat up the reaction to make the color change occur more quickly. That's number one. Can? Then, if I want it to go faster, then Bunsen burner lah. Even hotter what? Not even Okay, I keep waving. Teenager. Very close. Wrong word. Teenager is for what only? Very good. If I heat it up directly on a Bunsen burner, the sugars, if present, will decompose. So let's say I give you a test tube of glucose. If I boiling water bath, what color will I get with Benedict's? If a boiling water bath, a test tube of glucose will give me what color? Hey, hello. Brick red. Answer please. Okay, a test tube of glucose, Benedict solution, boiling water bath, what color? Brick red. However, if I don't water bath, I straight away Bunsen burner. The color I will get is blue. Then you're going to observe, you're going to write down blue, no reducing sugar. So what do we have? We have a false negative. It should, there are reducing sugars, but because you conducted the experiment wrongly, you got a wrong answer. Do you see that? Okay, so this is why must heat speed up. Why cannot eat too much? You may decompose the sugars if present. Can? Right? These two things must be in your head. You must understand that. Can? Okay. Moving on to the next page. Talking about the polysaccharides, this is something I shared earlier on. If you look at the shapes over here, they are all made up of hexagons. Okay, they are all made of the same things because all polysaccharides are made up of glucose. So why are they different things? Arranged differently. Okay, they're all arranged differently. So this kind of diagram, I need you all to be familiar with this. Very, very telling sign. When the question give you this kind of diagram, if all the individual shapes are the same shapes, carbohydrate already, immediately. Immediately. Why? Because what would the shape be like if I was looking at a protein? What are the individual things called for a protein? What are the individual things called for proteins? Amino acids. And in our syllabus, we learn how many different kinds of amino acids. You don't need to know what they are, but you need to give me a number. How many different amino acids are there? How many different amino acids are there? I need you all to dare to make mistakes. Say the answer. Wrong, we learn. Correct, we rejoice. What number? 3? Nope, more than that. Close, 20. Okay, there are 20 different amino acids. It is the way they come together, the how they come together, the how long they come together that forms the hundreds and thousands of different proteins in the world. The simplest example I can share with you is, how many letters are there in the alphabet? 26. How many words are there in the world? All right, now, why from 26 letters can have so many words? The way they come together, right? Exactly the same thing. 20 different amino acids, so many proteins in the world. How? The way they come together. Okay, coming back over here. This one got no difference. Only glucose molecules. That's why they're all going the same shape. Okay? Okay, let's go. Iodine test for starch is the first food test, in fact, that we have learned since primary school. And the unique thing now to highlight to you is that this is the only food test in your syllabus that can be done directly on a solid sample. All other food tests can only be conducted on an aqueous solution. Can? Right? The iodine test for starch is the only one that can be directly conducted on a solid sample. Now, let's move on to our next nutrient. Oh, wait, before we move on, Elizabeth had a question. Are polysaccharides sugars? I believe I said that just now. Are they? Are polysaccharides considered sugars? No, right? Elizabeth, we use the story of does white rice taste sweet? They don't, so not sugars. Okay, not sugars, And answer to your next question about difference between the differences and the similarities between polysaccharides are in this frame over here, Elizabeth. Okay. Okie dokie. Let's move on. We're looking to the next component of fats. Let's see how many of you have learned this in school already. All can tell me now. Fats and lipids. Which one is the bigger categorization? Fats or lipids? Which one is the bigger categorization? So in your syllabus, firstly, I want to assure you that you don't have to know this. In your syllabus, fats and lipids are interchangeable, but I want to share with you that lipids are the bigger classification. Okay? Case in point, if you look at this, major component of cell membranes. Our cell membranes are mostly made of a type of lipid. Okay, very, very important. I want to stress this because mainstream media have put a very bad reputation on lipids and fats nowadays because like, oh, fats are bad, right? Right. But please make sure we understand that if there is no life without fats, literally, cell membranes are mostly made of lipids. If you think about that, if we don't have enough lipids to make our cell membranes, there will not be any cells, and there will be no life. There will be no life. Okay, so gotta keep that in mind. All right. So for fats, the shape of the basic unit, I think most of us are going to understand it very easily. It's just going to be this sideways jellyfish, isn't it? Right? We have the glycerol backbone and the fatty acid tails. Right? The glycerol backbone and the fatty acid tails. Fairly straightforward. Right? This is only one type of fat. Okay, this is only one type of fat. Have you all been introduced the name of this fat molecule? Try something. Close. It's a triglyceride molecule. Okay, it's a triglyceride molecule. Right, we can pen this down, put it at the side over there. Just know that in your syllabus, fat molecule will do. Fat molecule will do, but there are other fat molecules. They have other shapes as well. Okay? Okay. So, ethanol emulsion test for fat. Now, this is a less commonly tested food test compared to Benedict's, definitely. Right? And because of that, right, most of us are not really very sure of this test. Now, let me explain to you how this works. I'm sure most of us, if not all of us, have had soups or like went for a steamboat. Then after a while, we see that the oil layer separates from the soup. Can we have the image in your mind first? The oil layer separates from the soup, right? Yes, yes, no, yes, shakes or not. Do you understand that? Thank you. Okay. So the reason basically is oil, it contains fats. Fats and water don't like each other. Simple as that. Fats and water do not like each other. So why does the ethanol emulsion test work? If oil and fats don't like each other, oil like who? Oil like ethanol. Oil likes ethanol. So I add ethanol first. If any oils are present, if any fats are present, they will stick together like good friends. Then I introduce them into water. So these fats and ethanol mixture will stay together but stay away from water. And that is where we have that separation of that white emulsion in that clear and colorless water. Okay, so this is how the ethanol emulsion test works. Right? Any oils present will befriend ethanol and stay away from the water. That is the ethanol emulsion test. And this, for this exact reason, we must ensure that we add the substances correctly. Ethanol first, filter, then water the residue. Okay? Right? So take 30 seconds, read these two steps properly. Take 30 seconds, read these two steps properly. If there's any confusion in your head, please let me know. And while you are doing so, for friends at home, if your Zoom name here put just initials like LX, if you put just a dot, I do not know who you are. Can you help me, do me a favor to change it to your name, please? Like IQLA, Ron, YX. Change it, please. Okay, change it to a name that I can recognize, please. Do me a favor. Thank you. Okay, any clarifications for ethanol emulsion? Okay, because I actually want to hit back here. Sorry, this one. This is why just now the carbohydrate, we need to know that H and O is 2:1 for carbohydrate. So if I have another molecule, also got carbon, hydrogen, oxygen only, but you count the hydrogen and oxygen, no ratio of H2O leh. What kind of molecule is this? It's a fat molecule. Can? Okay, so both carbohydrates and fats only got CHO, only got CHO. But carbohydrate got H2O, fats don't have H2O. Again, re-emphasizing the idea that fats don't like water. Okay? Okay, so use the same concept to help you understand many others. Okay, so carbohydrate, hydrate got water. Fats don't have water. That's why fats don't like water also. Okay, come. So with this in mind, having talked about the ethanol emulsion test for fat, I always like to ask this question. Listen carefully, please. If I suspect that this test tube got lipase, what food test should I conduct? If I suspect that a test tube got lipase, what food test should I conduct? Let's see. We'll find some names over here. Jiaxing. Who is Jiaxing? Hi, Jiaxing, come. If I suspect that I have a test tube that contains lipase, what food test should I conduct to confirm this suspicion? Why? So lipase is a what is lipase? Yes. Thank you for getting tricked, right? For some of you, lipase, ASE. It's an enzyme. Do you see that? The gears turning in your heads here. Lipases, just like amylases, just like your proteases, are enzymes. All enzymes are proteins. Enzymes are proteins, and therefore we should conduct the burette test. Okay? Do not get tricked. So thank you, Jiaxing, for getting tricked so that we can learn together here. All right? Okay. So speaking of proteins, over here, if you look at this, so carbohydrate and fats, CHO, straight up CHO. But the moment you have so many others, chomps, protein already lah. Straight up. In your syllabus, very simple. If you got more than CHO, protein. Okay? Now, the basic unit is an amino acid. The basic unit is an amino acid. And very fortunately for our syllabus, all we need to do when the question asks us to define or describe an amino acid, you just need to tell me it's a basic unit of a protein and contains an amino group and an acid group. Like quite literally, amino acid means you got amino acid group. Very straightforward. Okay? Very straightforward. Of the three biological molecules, carbohydrates, fats, and proteins, carbohydrates and fats, I'll classify them as resources. They are the things that get used up. Who uses them? What gets things done? So resources need to be used up by workers. Proteins are the workers. Proteins are the molecules that do things. And the most important thing that you have learned are your enzymes. All enzymes are proteins. They get the job done. Okay? They break down your carbohydrates or your fats. They synthesize your carbohydrates or your fats. Okay? Okay? All right, so just keep that in mind. A lot of things that they are doing, enzymes, antibodies, hormones, for movement as well. Okay? So the burette test is over here. I want to bring to your attention this phrase over here. So if I have a test tube of amino acids and I conduct the burette test, what color should I be expecting? A test tube of amino acids only. I do the burette test. What color should I be expecting? What color should I be expecting? Sorry? Why purple? Share lah, share. Wrong, we learn. Correct, then we cause I have a test of amino acid. It will cause burette test to turn purple because protein are amino acids proteins. How do we link those two terms together? Amino acids make up proteins. But this is a test for proteins. So you look at the phrase that we have just highlighted. The burette test actually only interacts with the bonds that are formed between the amino acids. Right? So that's why if it's just amino acids that are singly floating around in the solution, got no bond, no color change. And that's why it's a test for proteins and not a test for amino acids. Clear? Thank you. Okay, so gotta keep that in mind. If it's just a solution of amino acids, no color change. Right? Likewise, before we move on to enzymes, I want to talk about this one as well. If I have a test tube that I suspect contains lactose, right? If I have a test tube that I suspect contains lactose, I conduct Benedict's test, brick red. Can I conclude that lactose is present? Why not? Thank you. The Benedict test is for reducing sugars. Any 5 of them will give me the brick red. So how does this play into our understanding of the Benedict's test then? If the question says, suggest a test you will conduct to test for the presence of glucose, your first sentence must be glucose is a reducing sugar. If you just straight away say do reducing sugar test, why reducing? Maybe you forget that. Maybe you think that glucose is non-reducing sugar, how? Okay, if the question says test for the presence of glucose, you cannot do the Benedict test straight away. You must say first, glucose is reducing sugar. That's why you can do Benedict's test for reducing sugars. Okay? Does a test exist for glucose for the individual saccharides? Definitely, but we don't need to learn it lah. Can? Okay, so please keep that in mind. Thank you. Okay, enzymes. Moving on to enzymes. All right, enzymes. A very easy to get 2 marks just for the definition of an enzyme. Right over there. Okay, please keep this in mind. I would actually even suggest let's put this in over here. Biological catalysts that speed up the rate of chemical reactions by lowering the activation energy. And they remain chemically unchanged at the end of the reaction. Remain chemically unchanged at the end of the reaction. Now, remain chemically unchanged. To help you understand this story better here, just think about this. Hey, what if now I ask each and every one of you to shake hands with every other person over here? Do you need as many number of hands? Or can you just make do with your 2 hands? Can or not? After you shake a person's hand, you get back your hand. Your hand doesn't stay on that person. So you can keep on using that hand over there. Right? So remains chemically unchanged. Your hand remains as your hand in this particular shape. So after you've gone in for the shake, you grab the person's hand, you give it a good shake, you release it, you get your hand back into the exact same shape. Remains chemically unchanged. Alright? Okay? The next idea I want you to have in your head here is, when I just said, before I even brought my hand out, I say, shake another person's hand. In your head, you know that you're going to extend your hand. You're not going to go for the head or the leg or the stomach. You know that you're going to go for the other person's hand. So this means we're looking at the specificity. A handshake will always go to the same part. What does this tell me? Imagine if now we have this enzyme and substrate thing. These 2 parts are what to each other? Complementary. Thank you. They are complementary to each other. So think of the handshake now. Think of the handshake. Okay? Your hand is not going to be complementary to the other person's face. Okay? It's not going to be complementary to the other person's foot. Hopefully not. Alright? Alright, so it's going to be complementary only to the other person's palm. You're going to give it a good shake. Right? That's what we're looking for. Complementary. Now, lock and key hypothesis helps us to understand this whole complementary model kind of thing. The lock and key hypothesis is a model. Whenever the question asks, based on your understanding, based on your knowledge, using the lock and key hypothesis, explain how such is broken down by amylase, for example. Okay, the first sentence must always be to say which one is the enzyme, which one is the lock, which one is the key. So don't write this down first. Let me eyes on screen. I'm going to write this sentence down. Don't write down. So what is wrong? And is that, okay, firstly, have I identified my pairings correctly? Have I identified my pairings correctly? Yes or no? Come, nod and shake vigorously, please, so I can see where you stand. Please take a stand. Okay, yes, we have identified it correctly. Thank you for your nods. So enzyme lock. Why? Because you think about it, substrate enter active site of enzyme, key enters the lock. Fair? Okay, so first part correct already, but this sentence is incorrect. Having identified the correct pairing, this sentence is still incorrect. Please tell me why. Not yet, not yet.
It's starch. What is starch?
Okay, that one not yet. What is wrong with this? Just this one sentence. Not yet. What? No. What? Hey, hey, don't whisper to each other. Tell me, come. What were you thinking of? No lah, you talk to your friend, right? I thought you had an idea. The product. No, product not yet. Okay, then okay, see if I say like amylase is the wild starch. Still wrong. It's a very specific term thing. Not the wild. Yes, yes, yes. They are not log. They are not keys. The log and key hypothesis is a model to help you understand how it works. If you write this sentence down, no marks. Even though your head is like, yeah, what, log and key, what, enzyme log, substrate key. So how then do we use this sentence properly? Instead of is the logged, you can either say represents or XS. Fair? Can we agree with this? Yes? All right. Please do not say that they are locks and keys. Okay, you cannot secure any doors with them. All right, so please use represents or XS. Okay, represents or XS. Can? All right, so whenever the question asks us to using your knowledge, using based on log and key, always say this sentence first, state this sentence first. Then you go and talk about, oh, okay, complementary to the active side, breakdown to the product, so on and so forth. Can? All right, wonderful. Thank you for working on this with me. The next thing is, okay, this one very simple. Why does the substrate enter the active site of the enzyme? Why does it enter? Because they are complementary. It's very simple. Okay, substrate enters active site of enzyme because they are complementary. Okay, so everybody can understand, right? With that in mind, please keep it in your head. Tell me why the products exit the active site. Why would the products exit the active site then? Yes, it's very straightforward. If enter because complementary, that's it. It exits because they are no longer complementary. And this allows the enzyme to go on and catalyze the next reaction. Okay? Complementary, please come in. Oh, don't worry, catalyze you, no longer complementary. Please go. The next one that is complementary can come in. Okay? Enters because complementary, leaves because no longer complementary. Okay? Okay. So the next table over here, fairly straightforward. It's just about the names, right? So most enzymes are named after the substrate that they are going to break down. With one exception listed in the table over here, which is amylase and starch, isn't it? Why is it not called starches? Okay, so you will not be asked to explain this, but since it's here, let me just take this 1 minute to explain to you. Starch exists in two different forms, one of which is amylose, and therefore amylase breaks down amylose. Okay, if you're ever wondering why there's no starches, but for everything else in our syllabus, it is going to be named after its substrate. Okay, okay, everything will be named after its substrate. All right. So how are enzymes affected by temperature and pH? Are enzymes affected by temperature and pH? I am going to zoom my screen out so I can look at both graphs at the same time because the temperature graph is asymmetrical above the optimum. but the pH graph is symmetrical about the optimum. The next thing I want to do here is I'm going to use the same green arrows for all three sides like this. And then I'm going to now zoom in at the pH one. So the words I'm going to use here is when pH moves away from the optimum, the enzyme denatures. Away is bidirectional. Both directions away from the pH. So increase above pH optimum, decrease below optimum, the enzyme will denature already. What is this in contrast with? For temperature, only when temperature increases above optimum, then you have denaturation. Okay, then below optimum, how? What happens to the enzyme? They're inactive. They're inactive. Wonderful. Okay, inactive. The story I want you to have in your head here is think of the enzymes as just being asleep. If you are asleep, can I wake you back up to do your homework? I can. Okay, right? So likewise, an inactive enzyme, how do I reactivate it? I just have to increase temperature back up towards the optimum. Okay? Above optimum temperature, the enzymes are now denatured. The story in your head should be they're dead. So I cannot bring you back to life. Okay, denatured enzymes remain denatured. So if the question asks, hey, I change temperature from 40 to 60, then later I bring it back to 40, can still function or not? Cannot lah. But if I decrease to 10, then bring it back up to 40, can function? Can. That's it. Okay? Right? So I remember for temperature, both sides different, but for pH, both sides denatured. With this in mind, enzymes, therefore more sensitive to temperature, pH changes. pH changes. Okay? So fortunately, in our syllabus, we are required to explain more for temperature change. Right? So think about your energy changes one. Oh, more energy, break the bonds, lose the active site thing. Whereas for pH, you just say change pH, lose active site. Okay? Right? Further explanation will be available in our curator notes. So I see you next time as well. Okay? So in this case, okay, so for temperature, if they ask you about an enzymes and they say, oh, I changed temperature, blah, 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 we always assume first, if the question doesn't give you a specific scenario and they say, oh, this enzyme is amylase, you assume that amylase is from humans or just mammals in general, and therefore that enzyme's optimum temperature will be body temperature. What number should be in our heads now for body temperature? 37 degrees Celsius. Now, always have two temperatures in your head. Body, we think of 37 degrees Celsius. What is the other temperature we should have in our heads? Room temperature, which is 25 degrees Celsius. Okay? Okay, don't say, hey, climate change, then your room temperature change. These numbers that we set for room temperature, right, is not your classroom, your bedroom, or whatever. It's in a controlled laboratory setting where everything is like controlled very well. One. So we stick to the number of 25 and 35. Okay? Okay? Okay. Moving on. Before we move on to digestive system, any clarification required for biological molecules? Okay. So digestive system. I want to look at these two words. We're gonna write this thing down. What's the difference between these two sets of words? Sherry? Who's Sherry? Who's Sherry? Oh, hi, Sherry. Come. Can you tell me the difference, please? Should bring the let people try. Come, try. Come. What's the difference between the alimentary canal and the digestive system?
Um, where the food passes, like the food passes slowly, but the system is like how it gets digested.
Wonderful. I'll take it. Thank you. Right. The alimentary canal comprises of organs where food does enter and flow through. The digestive system is the bigger umbrella. The digestive system comprises organs of the alimentary canal and a couple more. What are the couple more? Food got pass through or not? Have undigested food pass through. Pancreas, liver, gallbladder. Yeah, the bile duct connects the gallbladder to the duodenum. Wonderful, thank you. So alimentary canal is only when you plus. Then it's part of the digestive system. Can you see that? You understand this Venn diagram thingy? Okay, digestive system comprises alimentary canal and these three other organs. These three other organs are incredibly important for digestion to occur as a whole, but food does not enter them. So they're not part of the alimentary canal. Okay? All right. So the way I did this tables over here is I pulled this out from the full set of the curator notes over here. So it should be fairly easy to understand already. I'm gonna move on to the more the points I want to highlight to you. Okay, esophagus. I always have fun with this question. Peristalsis. 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 Okay, aside from how to describe it, which is over there, listen to this sentence. Peristalsis occurs as long as there is a cylindrical muscular tube. So you can see, right? Esophagus is a cylindrical muscular tube, right? Okay. Where else does peristalsis occur then? Small intestine, large intestine. The stomach, some texts say peristalsis, then they put style, special form of peristalsis. It's just churning. The action of shaking. Why? Peristalsis is only when there are two muscles involved, the circular ones that squeeze like this, like circular, and the ones that run along the esophagus. Then it squeeze like this. That is peristalsis. Stomach got circular, longitudinal, and then diagonally running muscles as well, which is not in your syllabus. So stomach, don't say peristalsis, say churning. It's literally going nowhere. Stomach does this, but food doesn't go anywhere, isn't it? But the purpose of peristalsis is to move food. Can we see a difference there? Okay, wonderful. So, esophagus, small intestine, large intestine. There is one more. There's one more organ in which I want you to tell me peristalsis occurs in. Come, throw me some answers, please. You have an answer in your head? Say that, Brandon. Hey, come. I say before, hey, wrong answers, we learn. Correct answers, then you move on. How? Come, friends at home, please feel free to try as well. Try lah. What you say, Brandon?
You say anus.
Anus is just an opening. So no peristalsis. Thank you for trying. The rectum is just a storage bag. No peristalsis. Come, try. Cylindrical muscular organ. Come, think please. Think throughout your body. Where can there be a cylindrical muscular organ that moves things? Bile duct no need. It just allows bile to drip only. Amber, not stomach. Say stomach, not peristalsis. Stomach is churning. Blood vessels is cylindrical muscular, but no contractions. Blood vessels, as you'll see later on in the topic of transport in humans, merely serve to allow blood to flow through. Okay, thank you for trying. What else? Come. What? What do you say? Come. Are just tubes as well. That's a vast vessel, isn't it? Ah. Okay, it's correct for us to think outside of the topic already. Very good. This one more cylindrical muscular organ is no longer in the digestive system. A gland produces, not a duct. But they can sweat duct also not. It's a duct. It is not a gland, not a duct, not a vessel. Find another organ. There is a cylindrical muscular organ. Now, a very common answer I got was the windpipe as well. No. You take a deep breath now. Do you feel your neck? No, right? Your neck is not going like, yeah. So, thank you for trying. It's not the windpipe, it's not the trachea. What else? The eye? What about the eye? But I'm looking for peristalsis, eh. I'm looking for your eyeball. When you look at something like, like cartoon, I want to zoom at something. Good try, thank you. Come. Okay. Do I have any Sec 4s here? Don't have. Sec 4s will know this already by now. But your Sec 3, right? You have encountered this organ already. That's why I want you to guess. You have not been told this yet. But this organ you encountered before. You know this organ exists. Everybody does. Find the organ. There's a cylindrical muscular organ. It is muscular but not cylindrical. What did you say? That one is a tube only. Iota is vessel. Come. No, I need a tubular structure. Yes! Yes, Sherry! Again! The fallopian tube.
Oh, is it uterus?
It's the oviduct. When you all learn this, primary 5. Okay? Right? So the oviduct is also a cylindrical muscular organ. Peristalsis does occur over there. Okay? Right? So that's the interesting story. Next time I would love to continue sharing with you when I see you in class next time. But please keep this in mind. Peristalsis is not a digestion only thing. That is the moral of the story over here. Peristalsis is not a digestion only thing. What's the purpose of peristalsis? Move something. If you are in part of the alimentary canal, I'm moving food. If I'm in the oviduct, what am I moving? Make a guess. The ovum, thank you. Can? Not the sperm. Sperm cells have the... Please don't call it the tail. Can someone tell me? Flagellum, thank you. Okay, but again, sorry for next time we will talk about repro. Oviduct and fallopian tube are both the same thing, okay? All right, wonderful. Thank you for taking this detour with me. Let's head back over here. And before I move on from the esophagus, again, peristalsis is independent of gravity. So that's why if I hold you by your ankles and I flip you upside down, how does the foot move? Same direction. This is why when we talk about peristalsis, right, please don't say for the foot to move down. Always tell me the organ. The peristalsis in the esophagus moves food from the mouth to the stomach. Tell me the organs instead of to move down. Well, if you lie sideways, how? You move to the right. You go the other way, move to the left. Correct or not? Don't tell me up, down, left, right. Tell me from organ to organ. Then will never be wrong already. Okay? Can? It's independent of gravity, So, stomach, like I mentioned just now, churning, right? I do not want to use peristalsis. Purpose of peristalsis is to move substances from one region to the next. Stomach is just to mix. Can? Okay? Right, so please spend time to read through the functions of the stomach over there. We have the hydrochloric acid, we have the proteases as well. Okay? Pancreas. So on this page over here, we have the three organs where that food does not enter, but they're incredibly important for digestion to occur. Right? Very important for digestion to occur. So we can see that pancreatic secretions are enzymatic, so they will be affected by changes to temperature and pH. However, will bile be affected by temperature and pH changes? They will not. Why? That's it. Thank you. Bile is not enzymatic. If it's not enzymatic, it cannot denature, isn't it? There is no active site anyway. Okay? Right? So it's just a chemical reaction, but it's not enzymatic. Okay? Not enzymatic. So bile will not be affected. Right? What kind of questions can we think of when the question give you a table? Oh, I boil this thing, then I put acid to this thing, then I put plain water, distilled water to this thing. Will bile be affected by these changes? No. Bile will still be able to emulsify fat. Okay? Right? So with this in mind, look at this sentence here. If a person has to have his gallbladder surgically removed, will bile still be present in the small intestine? Very good, right? Because the production of bile is by the liver. If the liver is not affected, you will still have bile. So what's the function of gallbladder? We have it stored and concentrated. Now, key idea here is, without the gallbladder, bile will constantly just drip into the small intestine. Okay, bile will just constantly drip into the small intestine. But with a healthy gallbladder, bile will only be released when the body senses that, hey, you got eight fats. Okay, then I release bile to help you break down the fats there. Okay? All right. Moving on to the small intestine. I'm going to zoom in over here. We have these three words. Now, in these three words are just different sections of the same organ. They are just different sections of the small intestine. And in the order, it is duodenum, jejunum, and ileum. First, second, and third. However, in your syllabus, I don't know why the ministry say, okay, please teach your students to only know duodenum and ileum. Okay, so you just duodenum, jejunum, and ileum. Can? Okay, so duodenum is the shortest one of the entire 6 meters or so coiled up in all of your bodies right now. The duodenum is only 25 centimeters, but hey, 25 not like that lah. Like about 6 cm only. 25 maybe about like this. Okay. On this very short segment of 25 centimeters, it is in fact the where most of digestion occurs. Just this very short segment. So most of the time we consider after the duodenum, no more digestion, more or less digestion completed already. So then what happens next? In the small intestine, after digestion is complete, you have absorption. Okay, so therefore, if you look at the front part, majority of digestion, duodenum, absorption for jejunum and the ileum. Okay, absorption for jejunum and ileum. Yes.
Oh, there's not a kind of duodenum.
Your syllabus don't need to focus on that. Fair? Can? Okay, I need to emphasize there are a lot of things for us to learn in the world. If I teach you every single thing, you cannot do O-levels, right? Okay, wonderful. Thank you for asking me that question over there. Okay, so with this in mind, question, what are absorbed in the small intestine? What are absorbed in the small intestine? So many things to list, right? So can I suggest that we look at it in the other perspective instead? Everything that you eat will be absorbed as long as you can digest it. So what common food do we consume that we cannot digest? Common fiber from plant matter. Okay, cellulose, very good. So again, let's put some puzzle pieces together. Why can't we digest cellulose? This is an in-syllabus question, just that no one tell you specifically before. Because in plants digest themselves. What are you thinking of? You're thinking the correct things. Find the words. Yes. Tell me the answer. We cannot digest fiber because we cannot break down cellulose because we lack. We lack. Yes, say again. Cellulase. Cellulase. You say we lack cell wall? Cannot. Okay? You think about what you just said. No one has explicitly told you this before today, but is it within your syllabus? Just now I shared with you, right? Your enzyme names are always after their substrates. I just changed the last few letters into A, S, E. Okay? So who can cellulose ever be digested then? Are there organisms on Earth that can digest cellulose? Like what? Herbivores. Herbivores. Rabbit, cow, horse, all these. Can? They have cellulase in their digestive systems. Can? All right.
You said fiber is cannot be digested due to cellulose.
Fiber cannot be digested because fiber contains cellulose. We do not have cellulase. Okay? With that in mind, why some people cannot consume dairy products? They lack lactase. Can? Right? They don't have lactase. Why? Why these people so poor thing don't have lactase?
I don't know. That's too bad.
Okay. Firstly, all humans can produce lactase at birth. Slowly, as they grow up, they lose the ability to produce lactase. Then they become lactose intolerant. Can? Okay? So what can these people do? Very good, right? If they lactose intolerant to like the maximum degree, you take one milk already. But they still, wow, I love pizza. Please give me some ice cream. They can take some pills. And what would the pills contain? Lactase. Very good. Lactase. Right? Consume the lactase right before their meal so that afterwards they have their cheese buffet. They will not have a toilet buffet as well. Okay? Right? So these are some things that can occur as well. Right? You know why lactose intolerance is only for dairy milk from like cows? Because cows lactose. They have hooves. Okay. Because they have hooves. Okay, come on. All right, thanks for laughing at that joke over there. Just to keep it a little bit lighter, let's move on to large intestine. So, since primary school, we have always learned that the large intestine is where water mineral salts get absorbed, isn't it? Okay, so question. Small intestine, large intestine, who absorbs more water? Small or large? Answer is small. Why? Why? See, very simple. Very good. The easiest level 1 answer is small intestine longer. So pass by more osmosis can occur, right? Cannot. Okay. Then secondly, why the small intestine so good at absorbing? Okay, but
Tuition.
okay, we have number 1. Did I put it here? Okay, look at this. Okay. Okay, so do you see that I'm putting folds, villi, and microvilli all together in the table that follows? I leave my screen on a diagram here. You look at the table there. Can you see that folds, villi, and microvilli all are for the same feature? What is that feature? Thank you. Increases surface area to volume ratio. You see that in the table below? Okay. What's the difference between villi and microvilli? What's the difference between villi and microvilli? Villi is smaller, everybody says. Okay. Microvilli is smaller. Micro smaller. Okay. Microvilli are actually a part of a cell. Can we see cells with our naked eye? Can I? If you can, please let me know. Okay, right? Microvilli are so much smaller. Microvilli are so much smaller. Okay, so the next time you walk through the supermarket or the wet market, if you walk past the meats section, if you see intestines, look at them. If you see the bumps, those are villi. You cannot see microvilli. Okay, all right. For those who eat intestines, the thing that you can see are villi. Okay, the thing are the villi. Okay, small microvilli cannot see on microvilli. As you can see over here, are parts of a cell. Okay, okay, all right. So these features are for small intestine. Large intestine have or not to a much smaller extent. So not only is the small intestine longer than large intestine for absorption, the small intestine is also more efficient at absorbing. Okay, so small intestine absorbs more water than large intestine. Then what the large intestine do for water? That's why we have the word excess over here. Whatever water that you still need, but the small intestine had not have had the chance to absorb, large intestine absorbs. Okay? Okay, what happens if the large intestine is so efficient at its function? And then what happens? Constipation. If the large intestine too lazy, how? Diarrhea. Okay? Okay? Let's go. Okay, we talked about this one already. Go ahead and read it. Liver function. Liver function. I want to look at these two. I want to focus on these two today. Regulate blood glucose. Deaminate excess amino acids. Ooh, sorry. Okay. I want to talk on number 3 first. Deaminate. First and foremost, please spell it correctly. Please spell it correctly. What's this? This can mean to kill someone also. You deanimate, it means you make the person stop moving. Okay, so please make sure you spell it correctly. Alright? Alright, so to deaminate, again, another reason why we need to spell it correctly is because it quite literally tells me what it means. To remove the amine group from the amino acid. I remember just now we talked about amino acids are made of amino and acid something. To deaminate is to remove the amine portion. And right, the name itself tells me so much already. But it's for amino acids. Why here we can store carbohydrates, but this one must excrete. Yes, simply because amino acids, we cannot convert them to store into something else. We cannot store them directly as amino acids. So we have to remove it. Okay? So this is very important when next time, if I do see you again for the topic of excretion. A very incredible topic for us to learn because it's something that we don't think about very frequently, but we always do. What is our main form of excretion? Respiration. Respiration is a process there. You are thinking of, what is this? What am I doing? I'm breathing is different from respiration. I hope by now you all know the difference. But breathing is a mode of excretion. What else? Sweating as well. Very good. What else? Urination. So in the topic of excretion, I don't think anybody have done, right? Do really? Do? Have? What's the main organ for excretion? The kidneys. Okay, so for this one, just the two keywords here I want to bring to your attention, urine and kidneys. Okay. A very wonderful topic for us to talk about here because it is about how the ultrafiltration occurs, your selective reabsorption, you have your osmoregulation. Okay, so how it plays into the entire network with transport in humans, with respiration in humans. All right, okay, so please keep that in mind. The overlap between these two topics is at the liver. Okay, what the liver does for excess amino acids. Okay, so when you're revising for excretion, please have that in mind as well. Okay. All right, so blood glucose concentration. We are looking at glycogen there. I'm just gonna move on to this portion over here. Definitely the pancreas is involved as well because it is the pancreas that releases the two hormones, insulin and glucagon. Okay, focusing on this graph over here, we look at that green thing, you learn homeostasis yet? Some learn already, right? Come, let's define homeostasis. Yes, maintenance of. Okay, I like the last part over there, right? So homeostasis is the maintenance of the constant, stable internal environment when there are changes in the external environment. Correct? Okay. However, we need to be careful when we talk about external because changes can also come from internal. Correct or not? Okay, very good. Okay. The removal one is for excretion. Okay, right. So here is homeostasis. This healthy range here is otherwise what we know as the norm. Correct or not? I prefer to not use the word set point because set point makes us think a single value, but it is not. It is a range. So the story I can share with you again once more is, as you are roaming around your house, you move from the bedroom to the toilet to the sofa, all is peaceful at home. But the moment you try and touch the main door, want to go outside, your mother, Brenda, where are you going? Okay, so this is where changes will occur, right? So in this case, only when you have an increase above the norm, then the body say, hey, something is going wrong. I need to make some changes. Likewise, when you decrease below the norm, then changes need to occur. But as you fluctuate within the norm, move around in the house, nothing is going to happen. So always remember to reference the norm. Can always reference the norm. Right, that's for homeostasis. Right, very quickly over here, above the norm, we have insulin. Below is for glucagon. And then we are looking at the effects over here, right? So please spend some time to look at the effects on your own here. What we can ensure to do today in the limited time we have is just to make sure that we understand the concepts first. Above the norm, insulin. Below the norm, glucagon. If you're too high, I bring you down lah. If you're too low, I bring you back up. Can? Okay? That's it. Anybody confused about insulin and glucagon? Right? This is where we must ensure that our brains do not get fuddled with glucagon, glycogen, glycerol, glucose. Okay, please make sure you do not confuse all of these things. And galactose, sure, very good. We have 5 of these very similar sounding words here. Okay, I always like to say, our bio notes and questions here is a treasure trove of passwords. You have all these random words over there to make your passwords. Okay, all right. So basically these 2 chunks of text here is helping you to explain the graph previously. So let's move on to diabetes. Now, chronically elevated. Chronically elevated. What does chronically mean? Chronic, a long period of time. So long term, elevated, just high, right? Because I can show you after this or just now, whoever go and drink milk tea, 100% one, you confirm got high glucose concentration. But does that mean you have diabetes? Thankfully not. Okay? So it must be chronically, where it's just constantly high. Your body cannot bring it down. Thank you. Okay, chronically elevated must be long-term one because you and I, healthy people, when we drink one very, very sweet drink, right, our blood glucose will rise. Then what will happen? This lah. It's only when this cannot happen, then it just like this. Okay? Okay? Who is it? Who is it? Hello, Rayanne. Sorry about that. I wasn't looking at the participants panel over there. Okay, so having a look at this, type 1 and type 2, can you tell me which one is the more common one? Type 2? Do you agree with type 2? Type 2 is the more common one? Yes. Why? Why is type 2 diabetes the more common one? Happens later and become more common.
Anyone?
It can happen to anyone as long as. Very good. Okay, type 2 diabetes is a result of frequent excessive consumption. Frequent excessive consumption. All right. So not drink 1 cup only, diabetes immediately. No, it's when you keep on drinking and you keep on drinking a lot. All right. So frequency and amount, very important. Note that I'm using the word carbohydrate, not glucose. Why? Why I eat other carbohydrate will also result in high blood glucose. I told you answer just now already. Think back to what we went through today. Why I eat carbohydrates in general will lead to high blood glucose. Very good. All carbohydrates you consume will be eventually broken down into glucose, no matter what carbohydrate. Can? Alright, it's not just say then I just eat white rice lah. Also can what? Because starch broken down into glucose. Can? Alright? Alright. Okay, transport in humans. Transport in humans. Oh, Elizabeth, not quite true. All carbohydrates will be broken down into glucose. Okay, they are not glucose. That's wrong. Okay. All right, transport in humans. I want to start off with saying that this is a topic that I love a lot. Okay, right? Because it's learning about the heart, right? What makes us beat. Okay. So in this topic, there are three main components of the system. You have the heart, you have the blood, you have the blood vessels. Okay. Transport in humans, right? Actually, in primary school, we learn this as circulatory system, isn't it? Can you remember primary school circulatory system? Let me share with you right now that we have actually two different circulatory systems. The one that we are learning over here is actually called the very good cardiovascular system. Let's break the word down. Cardio referring to the heart. Very good. Vascular referring to vessels. Very good. The cardiovascular system quite simply explains to us how the heart uses a series of vessels to move blood around. Okay, cardiovascular. So cardiovascular system is how the heart uses vessels to move blood around. The second system you have heard of before already. In digestive system. Starts with letter L. Very good. It's not called the fat system. Very good. The lymphatic system. Thank you. Thank you. Lymphatic system. Okay, wait, got relationship one. Now, what does the lymphatic system do? You only need to know two things in your syllabus. The first one is what? What's your name? What's your name? Angel is what Angel said already. It is the transport of fats. It's not called the fat system. It's not like the whole thing fat inside. The lymphatic system, point number one, transports fats. Point number two. Brandon, what did you say just now? The lymphocyte thing sounds similar, right? What is a lymphocyte? White blood cell. In other words, the lymphatic systems is it's a highway for our white blood cells. The lymphatic system is a highway for our white blood cells. So, in the absence of infection, the white blood cells will primarily be in the lymphatic system. Only some will be patrolling your cardiovascular, right? So in a healthy person, right, within the normal range of white blood cell values, right, it should be on the lower end one. If a person has an elevated white blood cell count, what does that mean? If a person has an elevated white blood cell count, this person is fighting an infection. That's why got more white blood cells moving out around the body. Okay? So in your syllabus, you only need to know two things about the lymphatic system. Transports fats, transports white blood cells. Okay? Okay, so we're going to focus on the cardiovascular system. We're going to be focusing on the cardiovascular system. All right, let's talk about blood. For red blood cell, everybody know already. The thing that I want to focus on maybe is contains hemoglobin is the most important feature. Think about it. If red blood cell no hemoglobin, you got biconcave for what? If red blood cell no hemoglobin, you don't have mitochondria or no nucleus for what? Correct not? Right? Hemoglobin is what allows the red blood cell to even transport oxygen. Okay, moving on. White blood cells. Now, we have two over here. We have the phagocyte and lymphocyte. correct? Phagocyte and lymphocyte. This is two of the many different kinds of white blood cells. Okay, so very thankfully, we don't have to talk about the different kinds of white blood cells in our syllabus for now. We only need to know these two types, the phagocytes and the lymphocytes. Now, as the name suggests, phagocytes feed on pathogens. I use this similar sounding thing to help you remember. Phagocytes feed on pathogens. So it is this word over here, the ingest and engulf, they feed on. And right, so it is something that is bad, right? So if I eat something bad, then the bad thing gonna affect me, like. What would the phagocytes do next? Can we recall? Eat already, then the bad thing is inside of me, then I die how?
I don't know. I forgot the word.
Come, think about it, please. After I engulf, after I ingested something bad,
It's fine.
I will break it down. So I will ingest and digest. Can? So I ingest and digest. Phagocytosis is ingestion. I will ingest and digest. Where's my pen? Okay. Okay, ingest and digest. Lymphocytes, however, they don't do the killing themselves. They are like the archers at the battlefield. They stand very far back. They shoot arrows only. The arrows here are the antibodies. Okay, it's the antibodies themselves that kill the pathogens. Okay? Okay? Now, for red blood cell, very simple. Two words for its function, transport oxygen. Okay? White blood cell, two words for its function. How would I summarize the function of white blood cells? Both lymphocytes and phagocytes do what? Red blood cell transport oxygen. White blood cell do pathogens. Okay? Protect the body, right? Okay? Platelets. What are platelets for then? What are platelets for? Blood clotting, right? Blood clotting ensures that I don't let something come in, I don't let something go out. What do I not want to enter? Pathogens, isn't it? Okay, why do I not want to exit? Blood cell, red blood cells primarily. Okay, very good. All right, so for the entire cascade, please remember, got learn, right? All the thrombokinase stuff. Okay, please go and revise that. Please go and revise that. Next time, if you have time, I see you again. We can revise again for the flow chart to help you better see how the cascade works. Can? Okay. So what about plasma then? Simply put, plasma is just where everything is transported through. Okay, where everything is transported through. All right, very important for this. Okay. ABO blood group. Do you all know your cell type, your blood type? You know, right? Okay, why is it important to know your blood types? It's about blood clotting, right? Okay, no. Incorrect to say blood clotting. Agglutination. What is the difference between agglutination and blood clotting? What's the difference between clotting and agglutination? For agglutination and clumping, the end state is the same. The end state is the same. Red blood cells clump together. It is the stimulus, the starting point, and the entire process. Blood clotting will only be stimulated by platelets releasing what? Thrombokinase. Okay, blood clotting starts off initiated by thrombokinase. Agglutination is simply when complementary antigen and antibodies react. So herein, please take note, complementary is not a word that is just for enzymes. As long as this shape fits kind of similar to this shape, this is complementary already. Okay? So if some of us are confused about the antigen versus antibodies, where are they? What do they do? Right? If you look at the diagram over here, I believe it is very nicely visualized for us already. But if you need further help here, the story I can share with you here, if you listen to the word antigens here, maybe I can suggest you think of these as the agents guarding the body, the red blood cell. Right? So the antigens are the agents on the red blood cell, so they are always protecting the red blood cell. Okay, what are they protecting from again? Now, the antibodies are the busybodies floating around in the plasma. So only when you have complementary and the busybodies recognizes a friend, then they will start chatting in the street. More and more of them see each other. Hey, very long time no see. Then they start chatting. They start to clog up the street. In this case, the red blood cells start to clump up and clog up the blood vessel. Okay, so antigens are agents on the red blood cell. Antibodies are busybodies floating in your blood plasma. Okay, so we're looking at just two very simple things. All blood types can donate to and receive from their own blood type. Fair? Right? If you are A, you can donate to A, you can receive from A. Quite obvious, but right? But if you want to cross blood types now, universal donor is O. Universal recipient is AB. That's it. Okay? Just these three things to help you remember the entire matrix. Who can donate to who can receive from. Okay? All right, blood vessels, blood vessels. All right, we're done with blood already. Let's look at our vessels over here. A for artery, A for away. I believe this is something that everybody learns by already. A for artery, A for away. Okay, so one way is veins are the opposite, so it's just gonna be towards away, and opposite is towards. Or you can also use veins bring blood in to the heart. Okay, all right, two methods for you. Either way. But I want you to look at where I have zoomed into. Arteries and veins are both for transporting. That's all they do. Please never say, oh, pump blood or like move blood along. No, they are just there. You want to flow through me, you flow lah. If you can flow, you flow. That's it. Can? All right, arteries and veins merely transport blood. The key thing for capillaries, what word do we use then? What's the function of the capillaries? Capillaries allow for exchange to occur. Well done, Sherry. Okay, so we're looking for exchange. Looking for exchange. All right, allow to be exchanged. So we have transport, we have exchange. Now, very quickly, just to highlight to you some of the key differences. Why do arteries have thick walls? Cannot. Withstand blood pressure is wrong. High blood pressure. If it's not high, why must I withstand? Make sense? Okay? Likewise, why do arteries have narrow lumen? Why do arteries have narrow lumen? Like I said already, blood vessels only transport. You want to flow, you flow. I cannot do anything to increase the pressure. Something. High blood pressure again. Very good, Angel. So again, I'm trying to help us learn more by remembering less. It's always about high blood pressure. Okay? And the opposite is true. Simply, if the question asks you about the vein got thin because no need to withstand. Vein got wide lumen, that's why it is much lower now. Okay? What a beautiful sight over here. Okay? So everything over here is needed in your syllabus. Maybe except for the words superior and inferior. Okay, so maybe except for superior and inferior, but at this juncture, I want all of you to help me. Point to your hearts, please. Do you know where your hearts are? Point to your hearts, please. Point to your hearts. Point to your hearts. Okay. Okay, very good. Nobody point to the right side. Thank you. Okay, hearts on your left side. However, since primary school, we first learn to take the pledge, right? Put over your heart. Your heart is not on your left chest. Okay, I want you to find your midline. So it's where your sternum is. There's this bony structure over here. You have flesh in the chest, and then you have bone over here at the sternum. It's called a sternum here. You just need to take 1 to 2 finger spacing to the left. 1 to 2 finger spacing to the left. This is where your heart is. It's actually much more central than we used to think it was. Okay, right? So actually over here is where you can feel your heartbeat. You touch here, got nothing. This is your muscle already. Okay, so the heart is actually very, very central. Okay, heart is very, very central. Okay, all right. So in this case over here, if this is where the heart is, why got superior and inferior? Look at the arrows there. If the, yes, very good. Right? So blood needs to return to the heart from the top, return from the bottom. So we have inferior and superior vena cava. Superior and inferior doesn't mean one is better than the other. Just because you come from the brain, you're better. No such thing. It's just an anatomical term for position. Okay? Right? But like I said again, your syllabus only requires vena cava. So you do not need to pronounce superior or inferior. Okay? Next thing I want to ask. Look at this. Why got two pulmonary arteries?
Lung, right lung.
Very good. Left lung, right lung. Everybody know you got two lungs? They are not just one giant lung like that. It's left lung, right lung. Can you understand that? Okay? Brendan? It's left lung, right lung, separate. Okay? Question. Think about what I just asked you about the heart. One lung is slightly larger than the other. Which one is it? Right lung is slightly larger. Why? Left lung shares space with the heart. Okay? Okay? Okay, Alright. So, same answer for why got both sides of the pulmonary vein. Okay, they coming from each of the lungs. Clear? Okay? Right, because we have questions before, they don't ask you to label it, they ask you to label this. You die. What is this? Don't know what is this. It's still the pulmonary artery. Okay, and there's no need for you to label left pulmonary artery, right? No need. Just pulmonary artery will do, pulmonary vein will do. Okay? Okay, let's move on. Cardiac cycle graph. Yeah, another part that everybody loves looking at graphs, right? Okay, you love the cardiac cycle graph so much, isn't it? Okay, this is where I always love to help us understand this point better, because this graph thing over here, it is something that most students will like, die la graph. Okay, right? So if this is you, not to worry. Let me help you understand this a little bit better, as I have with the previous topics already. Firstly, okay, so over here we have our four valves. Okay, our four valves over here. The first thing you need to remember is, hey, this valve is between what two regions? Okay, so if I look at left side of the heart right now, aortic valve is between which two regions? Aortic valve is between the aorta, quite obviously. The name is there, isn't it? Aortic valve is between aorta and left ventricle. Left ventricle, thank you. Bicuspid valve then is between the left atrium and the left ventricle. Okay, first step is always remembering this valve is between what chambers. Okay, now let's have a look over here. If this graph is for left side of the heart, what is the name of this semilunar valve? Aortic valve lah. Okay, this is the aortic valve. Okay, question. What is the purpose? Why do valves need to close? Prevent backflow. But if the question now asks you specifically, why does the aortic valve need to close? You must tell me the direction. Okay, if the question say function of valve, okay, prevent backflow, you will get your valve. But if the question is accompanied with, why does the aortic valve need to close? If they specify the valve, you better specify the region. Fair? Okay, if they specify something means they also want you to specify something. Okay, so with some space, maybe from the previous page, we're going to write down from where to where. Let's see, let's see. Hashini. Who is Hashini? Hi, Hashini, come. Backflow, not black flow. Come, Hashini, prevent backflow of blood from the aortic valve is between what two regions? And however, over here we are writing aorta to left ventricle. Do we know why? Can you understand this sentence, please? This is a sentence that I think most of us are confused with. Why is this sentence correct? Yeah, I don't want it to come down into the ventricle. So I want to prevent blood flowing backwards from the aorta to the left ventricle. I want to prevent this. Can we understand the language here? Okay, please make sure you are aware of this. Okay, Brandon, now if I ask you, tricuspid valve closes to prevent the backflow of blood from. Very good. Okay. Can learn how to use this sentence. Now, what we have just written is actually the purpose. The purpose of valves closing. The other question that is very similar is the cause. Two very different questions here, isn't it? The purpose of valves closing and the cause of valves closing. The cause of valves closing. What results in the valves closing and opening? Very good. Very good, Brandon. Pressure differences. So this brings us again to the same first point. What are the regions there, isn't it? So for aortic valve to close, see if you all can understand this. Based on what Brandon told us about pressure already, how do we understand this inequality thingy? The aortic valve closes when pressure of the LV becomes lower than the pressure in the aorta. But why would this happen? Why would the pressure in the LV become lower than the aorta? Why is there less blood in the left ventricle? What is the left ventricle doing? Huh? See, you all use one hand, grip the other one. You contract. Do you think here feel more or less pressure? Yeah, lah. Contract is more pressure. Relax is less pressure. So therefore, pressure in the LV, when it becomes lower, is because your LV relax. Okay, LV relax, it contracted just now. When the LV contract, it pushed blood into aorta. Aortic valve open. But now after the LV contract, it has to relax. So then the blood will want to come back in, isn't it? The blood wants to come back in. It is prevented from doing so because of the aortic valve closing. Okay? So over here, LV pressure decrease because LV relax. What do I say over here? What do I say over here? Do I say that the aorta contracts? No. What is the aorta? What is the aorta? It's just a blood vessel. Blood vessels, I said this just now, right? Merely transports. Okay? So this one, you don't say anything. Just say left ventricle relaxes, causing pressure to decrease below aorta. Aortic valve closes to prevent backflow of blood. Okay? But if it is the tricuspid or bicuspid, tricuspid is between your right atrium and right ventricle, isn't it? So if it's a tricuspid or bicuspid, right? One side you say contract, the other side you say relax. You have to talk about both sides. But this one, only one of them is a chamber. Okay, only chambers can contract and relax. Okay, all right. But thank you for not saying contract and expand. Okay, expansion for heat, no longer. All right. And with that, if you can understand this properly, if you can properly utilize the cause and purpose in explaining valve opening and closing, you apply it to all four valves. Apply it to all four valves. Okay? Okay, apply to all four valves. That's it. Okay, I hope I made the graph a little bit easier for you to understand. Okay, last but not least. Last but not least, coronary heart disease. What is coronary? Heart. Wow, coronary heart disease. If coronary means heart disease. Okay, so the coronary vessels, we are focusing on the coronary arteries. But what do coronary arteries do? If arteries transport blood away from the heart, wow, coronary artery transport blood away from the heart to the heart. Okay, so therefore, we must ensure that we are specific. Look at the diagram properly. I'll zoom it in for you over here. Coronary arteries, where do they branch off from? Look at my diagram. The aorta. So the coronary arteries transport oxygenated blood from the aorta to the cardiac muscles or the heart muscles. Okay, so please make sure you pen this down correctly. You either say heart muscles, muscle cells, cardiac cells. Can. So what is coronary heart disease? You must specify. It is the coronary arteries that are going to be damaged. Right? But as with every other organ in the body, if the muscle cells don't receive enough oxygenated blood, what can the cells not undergo enough of? Aerobic respiration. If they cannot undergo enough aerobic respiration, they cannot release enough energy. Thank you. And therefore, well, understandably, they start to die, isn't it? And that is a heart attack. Okay? Always remember that. All right? Always remember that. So, narrow, blocked, this part, tell me why? What can result in the coronary arteries becoming narrowed or becoming blocked? But it's not just fats. Very good. All blood vessels. This can happen to all blood vessels. Just that when the question says coronary heart disease, you focus on coronary arteries. Okay? So if I were to draw you a cross section of a blood vessel, if this is a coronary artery, what is this space called? The lumen. Thank you. Okay? So now if I have some build up, what happens to the lumen? It becomes narrow, it is now smaller. Less blood can flow through, right? So then, do the heart muscles say, less blood, okay lah, chill lah, I don't contract and relax so hard. What will do? What happen next? The heart will now contract more forcefully, pumps harder and faster to compensate. Now I need to bring you what you need previously. But the heart is a muscle. As with all muscles, they will tire, eventually failing. Again, heart attack. Okay, so always keep this in mind. A couple of things here we're looking at. Oh, this is what we wrote down. Excess fat and cholesterol. Okay, again, it's about excess. It's not just you eat one time, we're happy. Okay. And that is it for the three topics over here. Again, many more things that I would love to share with you here, but we don't have time for that for this session over here. Right, just focusing on some of the things here. Many more things that I wanted to share. Right, for example, for this topic, I always draw a very, very nice diagram to help all of you realize how blood flows from which chamber to which chamber and then go on to what blood vessels and stuff like that. Okay? All right, so the next part is going to be our mock paper already. Let's take a short little break here first before we come back over here to review the mock paper. Right, so the washroom, if I believe I remember correctly, it's only one cubicle by the lift there. Right, so please take your turns to go over there. Maybe I'll give you all a 10 minutes break. By 10 minutes, we will start again. Okay, so if you need to, there's the dispenser outside. There's some snacks for you to get from the cubicles outside. I'll see all of you back here in 10 minutes. Okay, I'll see all of you back here in 10 minutes. Huh?
这个太累，这个真的你这个烦的。嗯，你说，那你想放到去，等一下你，我在想你，我是开始，大家想来这个，看一下，我觉得。I thought I was zombie. I don't say anything.
Okay, really. I don't even have something to say.
I can see, but
Yeah, how about cancer?
I think myself.
Thank you.
Well, you certain picture is meaningful. So I don't know. I'm buying master.
Ah,
Maybe we can have a colors. It's you go find the line down you select.
嗯，不是同一条，
你发现我说，
嗯，同一条那个是很多的，你都可以在那边看到那个你说那种特别是不一样，你不要本来给他，嗯嗯，我的天呐，啊，
为什么？
那方面比这个方面讲是吗？
啊，原来啊，不记得。
那他是你现在给我安排的呢，
啊，
还是会传去？可以看到一下那个听到的声音，你放好久了，嗯，
很长，然后他那个那个 Masih tadi.
What is that one?
Udah bisa.
I get going of actually all the time. Right? See that? You don't know. Going on to it. How are you gonna do? Think of to ensure that all of us they want is better. And we'll do it. You can't stop by taking reasons. Very clearly of it. And the next test. Thank you. Thank you.
I just want to try one brain I know food. Yeah,
I haven't caught your name right. Sorry, what's your name again? What's your name again? Leah. Oh, I sing also. Okay. And you are?
Misha.
What is it? Lisa?
MYH.
M-Y. Oh, I see it. Yes, thank you. Are we all back? I think we are. Yes. All right, yo. Welcome back from our break. Can I have all of you help me to input your MCQ answers into this form, please? Right. For those who have done MCQs, thank you for spending some time to do that. Please help me to fill in your MCQ answers into this form. Please help me to fill your MCQ answers. Okay, for our friends at home as well, if you have once you have done the MCQs, right? If you have had time to do that, please fill it in for me. No, never. I never put the marks. Yeah. But I know you all will look at it instead. So I know I have to focus on that. Okay, guys, you guys give you like 1 more minute to fill this in, please, including our friends at home. Okay, the reason I never put the marks in the hole there is because I don't want you all to just look at the marks there. Right? There's no point in doing that. Right, let's look at the questions themselves. So, once more, thank you for spending time to do some of them. Okay, so first one. Okay. Majority chose C. Right. I mean, you doesn't mean maybe you correct Brandon. Come, Brandon, you help me. What is W?
The back and of it. This new
What do you think? Looks smooth or rough?
also has a W.
Right. W is the smooth ER. Let's hear from Nicole. Nicole. Hi, Nicole. What is the function of the smooth ER? Very good. Okay. Smooth ER got many functions that we need to know of. One of them includes fat synthesis. We are also looking at carbohydrate metabolism. The smooth ER also functions as the liver of the cell. Detoxification of cell content. Wonderful. Okay. What is Z? What is Z? Z is the Golgi body or Golgi apparatus. Now, the reason I'm bringing both of these two organelles to your attention now is because structurally they are made of the same things. So what are they made of? Membranes. Remember just now, literally our cell, we are cell membranes in membranes in membranes. So both the smooth ER and the Golgi apparatus are made of membranes. The only slight difference is in their shape and their location. Okay, the Golgi body, they are the shape, flattened stacks of membranes. Whereas the smooth ER, network of membranes. Okay, do we see that? Do you see the difference there? So for smooth ER, we are looking at one network or series of membranes. While the Golgi body here, we are looking for flattened stack. It's a flattened stack of membrane. Okay. All right. So comparison number one is their shapes. Comparison number two is actually just for location. Very straightforward. Where will the smooth ER be found? Surrounding the nucleus. And then the Golgi. It's just all around. There's no specific location. Can you see that? Right? So structurally, they are made of membranes. It's just that. But it's because of their locations and their shapes that gives them different functions. Okay? So for Golgi over here, what is one word that comes to mind there for Golgi? What's the function of the Golgi apparatus, the Golgi body? Modification. Right? Modification. Okay, yeah. The online people, this QR code don't have meh? The paper? Okay, where is it? This one. Have I tried this? Tell me whether it's here. Yeah, if not, I move on. Okay. All right. Where was I? Okay, so in this case over here, we have the smooth endoplasmic reticulum and the Golgi body. What is X then? What is X? What is X? Okay, so we are either looking, okay, this one very low quality, very low quality picture. So is it going to be the rough ER or is it going to be the ribosome? Okay, at this point here, the diagram like very blur. So how are we going to put this, looking at this? Thankfully, MCQ, we can look at the options to guide ourselves to the answer. Okay, so therefore, what does this mean? When we are looking at open-ended question, you definitely will have a clearer image. Can? Thank you, Ronald. Okay. All right. So what is structure Y? So everybody should know. Nucleus. Okay. Now, X is the site where polysaccharides are synthesized. What are polysaccharides again? Starch, cellulose, glycogen. Okay, glycogen. These are carbohydrates, right? What I was trying to do is to trick you to polypeptides. Polypeptides are synthesized here, not polysaccharides. Okay, so doesn't matter whether this is rough ER or ribosome. Option 1 is incorrect already. Can? Right? Regardless of rough ER or ribosome, option 1 is incorrect because it is going to be for polypeptide. Can? But why would option 1, let's say I change this to polypeptide, why would X be correct even if I put rough ER? The word says the site. The site simply tells me where. Are proteins synthesized at the rough ER? The where is correct, but what synthesizes proteins? The ribosomes themselves. Do we see this difference over here? Ribosomes on the rough ER do the synthesis. So where is this synthesis occurring? Rough ER. Can we see that? Okay, make sure we have that language very, very nicely dialed in. Next one, Y controls all cellular activities. Straightforward. Nucleus definitely controls. Right, so let me cross this out first. Let me take over here. Z modifies substances made in Y. That's what the word just now we talked about Golgi apparatus here is a modification, isn't it? So Z modifies. True so far. Made in Y? No. No. What would the Golgi modify? Proteins. Right? Proteins are going to be made at the rough ER. Nucleus. Nucleus can make anything or not? Nucleus can make anything or not? Okay, this is something that you will encounter in the future. So therefore, let me share this with you first. Right? The nucleus synthesizes ribosome itself. Ribosomes are synthesized in the nucleus. Okay, ribosomes synthesize in the nucleus. Right, so immediately 3 is out as well, because this is not it. W, detoxification. SER, definitely. We talked about this. Looking at 2 and 3. And okay, 2 and 3. So sorry, 2 and 4. What am I saying? 2 and 4. Okay, so majority answer is correct. Well done. So for this question here, we are looking at whether or not we can trace the movement of the radioactivity over here. Okay, so radioactive amino acids, meaning they're going to be used for protein synthesis. Protein synthesis, if we were to do it as an open-ended, what is the first organelle we write down? If it's an open-ended, the radioactivity will first be found at the amino acids. Where will amino acids first be brought together? In the synthesis of a protein. The rough endoplasmic reticulum. Okay, rough endoplasmic reticulum, specifically the ribosomes, isn't it? Right, but over here we only have rough endoplasmic reticulum, so quite obviously we have to begin with C or D. Do we go the smooth ER or the Golgi next? Definitely the Golgi for modification. Okay, smooth ER, no. Smooth ER and rough ER, separate function. So question 2 answer is C. Majority answer as well. Thank you. For this question, before we look at the options, what is the state of this cell now? What is the state? Plasmalized. Thank you. So, again, this thing don't do. Look at my screen. Tell me. What are these three structures? Okay, don't do again, don't do I'm just gonna write down what I hear first. If this one is cell wall, and then we have cell membrane. Then orange leh. Again. If this is true, then what is this? This part over here is cytoplasm. Here is cytoplasm. So, excuse me. Then green one is what? Then green one is what? It's still the cell wall. Why am I bringing this to your attention? Remember just now we draw the thing, the plasmalized cell, right? Cell wall is a space. Always label the space for the cell wall. The cell membrane is this one. In a plasmalized cell, the descriptor for a plasmalized cell always is the cell membrane pulls away from the cell wall, isn't it? Right? Cell membrane pulls away from the cell wall. So you need to be able to label that, hey, this is the cell membrane. Isn't it? The cell membrane pulls away from the cell wall. Okay? Opposite of plasmalized is turgid, right? The cell swells and becomes turgid. So only in a turgid plant cell, again, don't have to draw this. Allow me to do this very quickly. So nucleus, vacuole, cell wall, cell membrane. Okay, why this one I only draw two lines? Why this one I only draw two lines here versus here got 1, 2, and 3? Because realistically speaking, in a turgid cell, the cell membrane is fully flushed to the cell wall, isn't it? But it doesn't make sense for you using a pencil to draw one line, two lines, three lines. They are overlapping. Make sense? Okay, so please be aware when we have a plasmalized cell, where the labels are. Okay? Okay, so for this question over here, for a plasmalized cell, means excess water entering or exiting? Exiting. Okay, exiting. So therefore, if I were to draw this thing, can someone tell me what this refers to? What is the name of the gradient? Water potential gradient. Thank you for not saying the word concentration. If I were to add this on, tell me what this is. Osmosis. Think of a definition of osmosis, please. No. Across the partially permeable membrane. Now, osmosis can only occur across the membrane, but diffusion does not need the membrane. Can occur across membrane, no? Can, but don't need. Whereas osmosis need. So this is what I'm having for you. So if we have an exit of water, we have cell, we have solution like this lah. Can? Okay. So for this question over here, we only have one. Plasmalized and flaccid. Alright, plasmalized and turgid are opposite things, right? So they cannot be together. Alright. This is your typical iodine test for starch. Iodine test for starch. But for this partially permeable membrane, look at the question. It tells us that inside blue-black, original color of starch is just going to be white. Original color of starch is just going to be white. Why would the inside turn blue-black then? Why the inside turn blue-black? This tells me that the iodine could enter. Why could iodine enter? For a artificial, for a synthetic, for a man-made membrane, only one feature controls movement of substances. What is it? What of the membrane? In what sense? For an artificial membrane, only one feature affects permeability. Very good. The size of the pores, which is therefore the size of the particles as well, isn't it? So if only iodine can go in, but here remains this brownish color, means starch could not exit. So therefore, starch was larger than the pores of the membrane. Starch could not move, only iodine can, and hence A. Okay? Oh wow. Yeah. Okay. We got majority right. Very good. Okay, let's move on. Unsweetened bread. So no sugar sprinkles on your bread. What is the primary nutrient in bread? More specifically, starch. Thank you. Like I shared just now about the rice example. First taste of white rice, not sweet. After a while of chewing, it will become sweet. Why? We have two answers here. Starch broken down into glucose, starch broken down into what? Maltose. Amylase breaks down starch into maltose, not directly into glucose. Okay, please keep that in mind. Right, so the reason for the sweetness in the mouth after breaking down your starch is from maltose due to amylase. 4 is A. Okay, 4 is A. All right, question 5. So we are looking at answer is B. And answer is B. Right, thankfully, A is not true. If not, we will forever taste it sweet. Okay? And thankfully not. If not, now we just mouth sweet already. Okay? Alright. Let's keep that in mind. Extract of saliva. What does saliva contain? Salivary amylase. Amylase is an enzyme, which are proteins, which will test positive with burette. Okay. Any fats? Any fats in saliva? Thankfully not. Any sugars in saliva? Definitely not. So we are looking at B. Okay. Okay, let's go very quickly. Question 7. Egg whites. At this point, you are expected to know the main nutrients in common food substances. Like rice and bread, you all know is starch. Egg white is what? Proteins. Gotta keep that in mind. Egg white are proteins. So therefore, we want to observe a clear solution more quickly. What can we do more quickly? If I add more egg whites, what am I doing? It will become slow because I have more things to break down, isn't it? Okay. If I don't stir it now, become even slower as well, isn't it? Right? Because I have no stirring, just like how when you all make your Milo, you must stir, not the powder will just stay there. Okay? So A and B both will make it slower. Between C and D, yeah, these are our two majority answers. Okay? Majority chose the correct answer of lowering pH. Why will lowering pH work? Why will lowering pH work? Gastric enzyme. What's the pH? Gastric means what organ? Stomach. Stomach what? Hydrochloric acid. So I lower it towards the optimum pH of 2, I will increase the rate of reaction. Okay? D is incorrect because denature. Right? This one, if they never state, tell you already, gastric, your stomach, your body temp is 37. 65 is way too high. And denature. Excellent. Let's go. Question 8. So again, if you did not understand this properly, right, the entire agar plate contains starch. Okay, the entire agar plate contains starch. I dug a few circles. I put some seeds in, boiled versus unboiled. Afterwards, I removed the seeds. I added iodine solution, and these are the colors I observed. Obviously, with iodine and the entire starch thingy, blue-black, right, means presence of starch. Question is asking, hey, why in these places no starch anymore? Did the seeds eat up the starch? No, what is going to happen here? Question 8. Let's see, did we choose the majority answer? Oh yes, very good. D, right? The seeds secreted amylase. But why do seeds do that? Why would the seeds contain amylase to break down starch?
Usually the seed has amylase to break down like the food that they have or germinate.
Very good. Seeds themselves need to germinate, right? They need to break down their own stored food. Everyone learns this in primary school, right? Stored food for them to germinate. So therefore, in this case, the amylase just leaked out. Well done. Okay? Oh, cholecystectomy. Okay? Bio tongue twisters. Cholecystectomy. Again, like I said, don't need to be afraid about this kind of big words one. Whenever we want to ask you something that is not explicitly in your syllabus, we will tell you what happens. For all you know, right, the question will remain the same if I just say procedure X. Correct? Okay, so cholecystectomy here. When I remove the gallbladder, what can be expected after the surgery? What will happen to a person without a gallbladder? Can release bowel or not? Can. But how? What is glycerol? What is glycerol? The breakdown products of fat. Remember the sideways jellyfish? Glycerol is the oval, but nothing to do with this one. Okay? So number one is out. Lowered amino acid absorption. Anything to do with proteins and amino acids? No, absolutely not. Less bile is produced. The word here is produced. The liver is not harmed. The liver continues producing the same amount of bile. We are looking at 4 only. We're looking at 4 only because if there is no gallbladder to know when to release bile, the liver will just keep on dripping bile into the duodenum. Then the next time when you eat fats, right, you cannot like one shot. Let's put more bile all at once. It will always be that small amount only. Okay? Okay, question 9, we're looking at D, Oh, something should be fairly straightforward. Question 10. Yes, malnutrition. Okay, question 10 is malnutrition, Question 10 is malnutrition. Okay, because if it becomes smoother, you lose the increased surface area to volume ratio. Let's move on very quickly. Can we identify the vessels here, please? Identify the vessels here, please. We are looking. Oh, Elizabeth asked question 6. Yes, B. Okay. Okay. All right. Yeah. All right. How will blood in vessels E and F differ after a heavy meal of rice and potatoes? Again, once more, what is the main nutrient in rice and potatoes? Starch. Okay. So, blood vessel E. What is blood vessel E? Hepatic vein. Blood vessel F. Then the hepatic portal vein. Okay. Blood vessel F is then the hepatic portal vein. After you eat a lot of starch, all the starch you consumed will eventually be broken down into glucose, absorbed into the bloodstream. Where will this nutrient-rich blood be transported to? From the small intestine, it will always go to the liver first. Okay, so therefore, after a meal that is heavy in starch, you have a lot of glucose here. It flows into the liver. The liver say, hey, why you have so much glucose? Don't need. Let me store some for you. And hence, E will have less glucose than F. Answer is B. Okay? Any clarifications? Please let me know. Question 12. Can I first ask what does the systemic circuit mean? What does the systemic circuit exclude? The lungs. Very good. What's the name of the circuit that involves the lungs? Pulmonary circuit. All right, so double circulation in humans, isn't it? We have pulmonary circuit, systemic circuit. Pulmonary is for lung. Systemic, all your organ systems. Okay, all right. I just saw a question. Madeline asked, iodine, why is it not osmosis? Osmosis is only for water, no other way. Okay. Okay, so basically at the end, right, I will show you all the I'll pass you the answer key later on, so don't need to worry too much about it. Okay. What is the difference between 2 and 1 then? What's the difference between 2 and 1? What's the difference between 2 and 1? 2 is the coronary artery, 1 is the aorta. Right? How would you look at it? 1 branches off into different arteries. Can all your arteries branch off from the aorta? All right. Okay, so for question 12, we are looking at coronary arteries, and then we have the hepatic portal vein. And answer is C. All right, answer is C. Majority answer again. Okay, majority answer again. Well done. Question 13. See whether or not we can properly remember the valve things over here. Let's identify the valves here first. X is the... How do you identify X? X is the tricuspid. X is the tricuspid. Y? Y is the aortic. Wonderful, thank you. And Z has to be the bicuspid. Okay, Z has to be the bicuspid. What happens when the atrial contract? The atrial contract to do what? Very good, to push blood into the ventricle. So therefore, these two arrows occur. So if you look at these two arrows, the valves in which these two arrows pass through must open, right? So immediately we see that X and Z have to open. When the atria contract, the ventricles will relax. Hence, if the ventricular relax, ventricular pressure is high or low? Ventricles relax, ventricular pressure high or low? Low. So therefore, they have to close. Okay? All right, they have to close. If left ventricular pressure low, then open, then blood will backflow already. Okay? Okay, for 14 is about respiration here, fairly straightforward. We are looking at of the entire air sample, which one is relevant for candle to burn? Oxygen, definitely. Okay, we can see that. This one take less time, this one take more time. If you could burn for longer, you had more or less oxygen? More oxygen, right? So gas jar 2 contained more oxygen. So when comparing inhaled and exhaled air, which one got more oxygen? Inhaled air, because I need you all to understand, inhaled equals atmospheric. Inhale means literally, now if I then I put a needle here, I extract the air from inside my trachea, it will have the same, more or less the same composition as the air outside. Composition will only change at the alveoli, at the air sacs. And so inhale equals to atmospheric. Right? So therefore, if 2 had more oxygen, 2 has to be inhaled air. Right? So for this one here, 1 will be exhaled. So 14 is B. Okay? Oh, 15, quite an even split. Quite an even split between C and D. Now, think back to the last time you had your vigorous activity, your 2.4, or you chased after the bus. The moment you stop running, you're still panting, right? Why are you still panting? Why do you still have the increased breathing rate? You're still taking in more oxygen, but is it for the energy release? No need. You're trying to repay the oxygen debt now. But the repayment of the oxygen debt only occurs after exercise. Answer is D. Can? Right? While you are exercising, the body is like, well, oxygen, please give me aerobic respiration. No oxygen left for a repayment of oxygen debt. Can? Alcohol's effect on ADH production. Where is ADH produced? Okay, this one is where we have that liver gallbladder relationship. The liver gallbladder relationship is where the liver produces bile. The gallbladder is the one that stores and later release, right? Okay, not hypotenuse, nobody say hypotenuse, okay. Hypothalamus produces ADH, which is then stored and subsequently released in the pituitary gland. Okay, pituitary gland. But both of these organs are in the brain. Okay, at the base of the brain. Okay? Okay, ADH causes, this one, please remember, more water to be reabsorbed. More ADH, more water reabsorbed. Okay. Oh, wait. I had that wrong thing. Sorry. Wait. ADH causes more water to be reabsorbed. But the question is asking effect of such drinks which reduce. So therefore it has to be decreased. And question 16, we're looking at the decrease. So if I reabsorb less, question, water in the tubule reabsorbed into when? Into the bloodstream, right? So now the blood water potential go back up. Reabsorb into bloodstream. Reabsorb from, what is this thing called? What is this fluid called?
Blood.
Glomerular filtrate. Thank you. So water is reabsorbed from glomerular filtrate into the bloodstream. So now glomerular filtrate, less water, right? What will glomerular filtrate eventually become? Urine. Okay, glomerular filtrate eventually becomes urine. Glomerular filtrate, less water. You will have less urine. Less urine, there's more concentrated. Right? Oh, wait. I confused myself again. Wait. Right? But because of alcohol's effect, you cannot absorb as much. So therefore, you have more urine that is diluted. Can? Okay? Wow, even I keep confusing myself for this question. Okay? Can you understand this one? Anybody needs me to explain again? Awesome. Once more, the kidney. Oh, quite a mixed bag. Hey, so far, you're like almost every option get chosen. MCQ, every option gets chosen. Okay, come, let's move on. Question 17. Based on the arrows, can you identify blood vessel Q? Yes, we know artery is P, we know vein is Q. Alright? Q versus P. Q has more or less carbon dioxide. It's coming out from an organ. So there needs to be more carbon dioxide. Right? Okay, so more carbon dioxide. Water leh? More or less water. It has to have less water, isn't it? Glucose. More or less. More glucose means the kidney give the blood glucose. Why is it less? Sorry? Kidney made of living cells, right? What do living cells take from blood? Oxygen and glucose for aerobic respiration. Do you see that? Okay, so there will be less glucose exiting because the kidney cells themselves use oxygen and glucose. Can? Yes.
Shouldn't it have more because it's the percentage of glucose, not the amount of glucose.
So the solvent decrease, but your solid also decrease. Fair? Okay. All right, come. Homeostasis falls below 37 degrees Celsius. Which of these few features help him warm up? When blood rushes to the skin surface, what happens? Lose more heat, isn't it? Okay, so one is out. Drying the skin quickly, how? What does that help with? Less evaporation, less heat will be lost, okay? Hair erector muscles relaxing. What do the hair erector muscles do when they contract? Very good. Traps air that is an insulator of heat, isn't it? Okay. So when the hair erector muscle contracts, right, we are raising the hairs that traps a thicker layer of insulating air. Right this very moment, a very thin layer is being trapped. As long as there's an outer covering of hair, there will be air being trapped over there. Just a very thin layer. When hair stands out higher, a thicker layer will be trapped, more insulating. However, this effect is almost useless in humans because our hair is just too sparse, too soft. Okay? Alright? Relaxing will not help because it should contract anyway. Okay, next one, shivering. Will shivering help? What is shivering? One word is missing. Must be a rapid or vigorous contraction and relaxation of the muscles. If you don't say rapid, you just contract and then just one time only like that where got shiver? Okay, not? Okay, it's a contraction and relaxation, the very rapid one that generates heat there. Okay, shivering does running around help? Well, you shouldn't, but does it? Because it generates heat there, right? Okay, so we're looking at 2, 4, and 5. Okay, running around. Okay, controlled by homeostasis, an excellent question to ask. Which ones are controlled by homeostasis? Blood glucose, definitely. We talked about insulin and glucagon, isn't it? Insulin and glucagon. Okay. Water content in the urine. Yes or no? Okay, we have some nods. Okay, then this one, wait. Temperature in the liver. Yes or no? That was internal body temperature. It's a yes. Okay, 19 is C. How do you understand why 19 is C? Question asked, which ones are controlled by homeostasis? Does our body look at the amount of water in the urine and say, hey, urine not enough water, let me give you more water? Does the body look at the urine and say, hey, so much water, let me take back some? All right, we are not controlling water content in urine. What are we trying to control? Blood water potential. That is what we are focusing on. Okay? We are using urine to regulate blood water potential, not the other way around. Okay? All right, don't get tricked by this. Similar question as the above one for the homeostasis thing here, for the thermoregulation. Rises above normal, blood vessels near the skin will. What do I call this effect? This term you will only use for this topic. Vaso what? You want to widen. Vasodilation. Very good. And the opposite is vasoconstriction. Thank you. Right? This is the only time you use these two words. Okay. All other times you will just say dilate and constrict. Okay. Glands in the skin release. Well, glands in the skin do release sweat and oils, but which one is important for thermoregulation? Definitely sweat. Actions are controlled by the hypothalamus. Nothing to choose here. And body cools when water evaporates. And hence I'll say it's D. Okay. Okay, come. Let's move on to our open-ended questions now. For this one, again, like I said, let's look at some of the question analysis portion. Okay, the question analysis here, I want to look at if we have temperature of water bath, we're looking at the effect of temperature on enzymatic action. We are also therefore looking at the effect of temperature over here. Right? Okay, so even if the experiment was done at body temperature, near body temperature, but if I had already boiled the saliva, denaturation cannot reverse, right? It cannot bring you back to life. Therefore, there'll be no effect over here. And this is our odd one out. We need to be able to identify that this is flask R. Okay? Is that fair? Right? Now, 1 and 2, graphs 1 and 2 both reach 0. Graphs 1 and 2 both reach 0. Which one can starch be completely digested in? Which one can starch be completely digested in? We are looking at P and Q because they are both conducted at the optimal temperature. All right. Whereas we are looking at 3 here, going on so slowly, so slowly. This is the one where the enzyme is going to be inactive. And very, very inactive. It is the one that was less enzymes, lower temperature, very slow rate of reaction. Can? Now, I have starch, I have saliva, and I added hydrochloric acid. Which curve will be the same as flask T now? I add hydrochloric acid to this. Which curve will I get? I hope we all can see that it's going to be 4. Hydrochloric acid will denature salivary amylase. And hydrochloric acid will denature salivary amylase. So what are some of the points that we need to have in our heads here? If hydrochloric acid denatures our salivary amylase for 3 marks, the first part is to state that salivary amylase will be denatured by hydrochloric acid. How do we describe denaturation then? What happens to the enzyme? It's denatured. Right, we are losing the unique shape of the what of the enzyme? Not the entire enzyme. Active site of the enzyme. Very good. Okay, so that's your second marking point. So having denatured, what is next?
The shape is not complementary.
Very good. Bring in the complementary thing again. Okay, no longer complementary, cannot catalyze the breakdown. Okay, so 3 marking points, very straightforward. Okay, I'll show you the answer key again later on, so just make sure you have the points in your head. Talk about denaturation, talk about loss of active site, talk about no longer complementary. These are your 3 points. Okay? Moving on to something that we kind of talked about a little bit earlier on, we have oil stains, and like I shared earlier on, we need to know the main biological molecules in common food substances. Oil contains fats. Okay, so here, biological washing powder, very special. What is the special thing that helps remove oil stains? You need to tell me it's going to be lipase over here. Okay, lipase over here. An acceptable but less preferred answer is just enzyme. Okay, if you can specify the enzyme, do so. It's going to be lipase. All right. Then Eunice say, all right, increase to 90 degrees Celsius, more effective, yes or no? Definitely not. What will happen? Again, the whole denaturation thing, isn't it? Okay, so it's not very straightforward. We all know this already. Okay, we all know this already. What I want to talk about is part B. Test for the substance in A part 1. What is A part 1? What do you write down here? Lipase, right? Lipase, Then here, what test? Burette, very good. Okay, don't get tricked, What you're trying to identify lipase, so it's about the Burette's test. Okay? So in this case here, if you immediately started by saying conduct burette test, add the burette solution, see the violet, you will only get 2 marks. Why? Very good. You must first tell me. I asked you test for the thing, but you never tell me what the thing is. Your first sentence has to be lipase is an enzyme, which is a protein. Then you can use the Burette's test. Okay? Well done. Question 23. Where is the pancreas? The leaf thing, fair enough. It is the one right below the stomach. Many different diagrams will show slightly different variations. You just need to know what the other organs look like. And pancreas is the only one left. Okay? All right. So question asks, how the diet must change if the pancreas was removed? The pancreas has 2 functions. One for digestion, the other one for regulating blood glucose. This question, what do you think this question wants you to focus on? Okay, so this question here, we are focusing on the function of the pancreas. How will the diet have to change? This person must eat less or more of something. So therefore, if you want to focus on the Regulation part, you must specify about carbohydrates and then explain about the release of insulin. If you want to talk about the release of, the digestive function, then you're going to talk about, oh, okay, what kind of enzymes are released by the pancreas, and therefore, oh, less of these enzymes, less digestion can occur. Okay, two ways to look at it, as long as you can explain it properly. Okay, we are moving away from primary school. Last time, primary school, you all learn is you must give me this sentence. No such thing. Give me the correct idea. If you can give me the correct idea, you will get your mark. Okay? Yes, Nicole. And therefore? Okay, so very good point there. Nicole says, hey, then pancreas release both insulin and glucagon. How do I find the problem? Okay, I actually was much rather trying to guide you all to diet meat. And then the question say digestive system, focus on the digestive function of the pancreas. Most of the time, this kind of question, no pancreas, how? Talk about the digestive function. Can? Okay, wonderful point, Nico. Thank you for noticing that. Right, this question is always about first statement, digestive system. Picture, digestive system. Talk about the digestive function more. Okay? Okay? Identification of the heart, fairly straightforward. Please go ahead and slowly do that in your own time. But for part B, look at that. Before today, anybody see these two words before? Aortic regurgitation. Okay, so what's the point here? Learning point. Again, just now, cholecystectomy, removal of gallbladder. Aortic regurgitation. Does the word matter? Does the name matter? Doesn't, as long as I explain to you what it means. Can? Okay, so let's learning point here. Don't care about the words. Don't care about the name. Look at what they describe to you. Right? It's the aortic valve weakens and cannot function properly. We wrote down just now, isn't it? What's the function of the aortic valve? Closest to? Continue, please. Aortic valve closest to prevent backflow from aorta to LV. Always have that in your head, just like this question. Hey, no pancreas, how? Release digestive enzymes. So over here, aortic valve cannot function properly. How? Okay, first question. Volume of blood flowing through the aorta. Now I'm going to do some scribbles. Don't follow. Okay. What is this part called first? Left ventricle. The left ventricle is the one that contracts to force blood to the aorta, isn't it? Okay. So now when LV contracts, it's going to force, let's say, 10 units of blood into the aorta. The next moment, when the LV relaxes, what will happen? These 10 units will want to return, isn't it? But can it? These 10 units will be stopped by the aortic valve. So how much blood will then continue to exit to the rest of the body? 10 law. Can? Now in this patient, weakened aortic valve cannot function properly. How much blood forced out? Still 10. The LV, nothing wrong. Do we realize that? The LV, nothing wrong, still 10 units forced out. Then when the LV relaxes, 10 units want to go back. Okay, it's not that it's not there, the aortic valve, it just weakened. So maybe 2 units come back. So then now, how much blood? Can we see that? Okay, the numbers is just for me to help you understand only. Your answer, please go and say, assume 10 units. Okay, just say, allows some backflow. Hence, volume of blood exiting is smaller volume. Can? Do you see that? First part of the answer is just a smaller volume of blood flows through the aorta. How does this affect the body now? Less oxygenated blood, less oxygen and glucose. Then talk about respiration, and finally energy. You always link these three things together. And therefore, feel more tired, feel more lethargic, fainting spells, all these kind of things. Can? As long as you give me the energy one. Can? We talked about 2 ways arteries are adapted for their functions. Do we remember? The lumen is very narrow. What is that for? Maintain. Narrow lumen is maintained. Withstand is for? The thick walls. Okay, the thick walls. Okay, right? Thick walls is for withstand high blood pressure. Narrow lumen is for maintain high blood pressure. Can? Okay, is there another possible one? What about the elasticity? When? Correct, correct. Okay, so the arteries have to be elastic. I want you to imagine now, this is the artery lumen, for example. When a very high blood pressure unit of blood pressure flows through, right, what's going to happen is going to actually punch into the artery. But look at what happens to the lumen size here. It will have to widen to allow the unit of blood to flow through. And it's actually this widening, right, that you feel when you're looking for your pulse. It is literally the blood punching at the artery walls that you are feeling as your pulse. So that is for the elastic recall as well. Well done, Nicole. Can? Okay? And this one, semilunar valves. Have we talked about the graph earlier on? Right? It's about the cause or purpose. This question here, are they asking about cause or purpose? This is about cause. So therefore, we talk about pressure. Simply put, first close. Why were the semilunar valves closed? It's literally the same thing we talked about, right? You want to prevent backflow into the ventricle. So when will they first close? When the ventricles start to relax. How do I know whether the ventricle is relaxing or not? From this graph, what about the pressure? When left ventricle relaxes, pressure will drop. So therefore, this is the only part where LV drops, ma. So answer is C. Okay? I identify a point C first, isn't it? Okay? Explanation is all that pressure thing, Can? Right? Go refer to what you looked at just now. Can? And then this one, patient with blood type B. Describe the importance of donor-recipient matching. Quite a general question here. What kind of marking points are required? Definitely, we're talking about agglutination. If the patient is a blood type B, what antigens does he or she have? Antigen B. Antigens are on the red blood cell. It is the antigens that tell us our blood type. So blood type B means antigen B on the red blood cell. What antibodies does the patient have? A. Okay, that is your first marking point already, immediately identifying the antigen and antibody pairing in this person. Okay, so importance of donor recipient matching. Hence, you have to tell me if wrong, how. So choose any other blood type. This one cannot give to this patient. What blood type is this? A. Right? So cannot give A because when you're donating blood, right? I need you all to understand is it the red blood cell or is it the plasma that I'm trying to donate? Red blood cell, because what is plasma? Just water with all the dissolved things, right? So I can make what? Alright not? Do you realize that? Plasma can make one. Red blood cell is the one we cannot make. So red blood cell is the one that we're trying to donate. So why cannot give red blood cell A to a person with blood type B?
Antibody A in the blood plasma of the person.
Okay, so blood type B person got antibody A will react with antigen A of the other person's red blood cell. Okay? What is the relationship of these two, antigen and antibody? Complementary. Can? So you talk about the agglutination there. Fair? Okay? Then you must, so the identification is of the antigen versus antibody. What am I donating? Focus on the red blood cell. Okay? Focus on the red blood cell. All right? Okay.